Hello, my name is Joshua Gavi. I welcome you to Reproduction in Living Organisms. Questions 31, 35. Outline the events which lead to fertilization of an egg by a sperm. Part B. Outline extraembryonic membranes associated with human fetus. Outline the events which lead to fertilization of an egg cell with a sperm. On entering the vagina, sperm spends about 7 hours being capacitated, after which they move towards the oviduct, aided by the muscular contractions of the uterus and the oviduct, and the lashing of the tail. A spermatozoon comes in contact with the oocyte by random movement. So when you look here, you'll see our sperm moving towards the egg, but it takes some time being capacitated, and after that one it moves towards the oviduct, aided by the contraction of the uterus and the oviduct and its lashing tail. Then it comes into contact with the oocyte. Oocyte is the egg cell. Acrosome enzymes, they hydrolyze the granulosa layers of the egg until the sperm head makes contact with the zona pellucida. Sperm acrosome membrane ruptures and releases the hydrolytic enzyme acrosome reaction, and the acrosome filaments pierces through the oocyte membrane to the plasma membrane of the oocyte. So when the acrosome enzymes are released, or they pass through the granulosa layer. The granulosa layer is the outside layer of the oocyte, and when they pass through the granulosa layer, the sperm cell comes into contact with the zona pellucida, this layer here, indicated by the green color, zona pellucida. And when it touches the zona pellucida, the sperm acrosome membrane ruptures and releases the hydrolytic enzymes. Those hydrolytic enzymes now will hydrolyze or break the plasma membrane and the oocyte membrane to allow the sperm nuclei to enter into the oocyte. An electrical potential change in the oocyte membrane occurs, what we call the first block, followed by fusion of the cortical granules with the plasma membrane to discharge their content what we call the cortical reaction. And this creates an osmotic gradient that draws water into the space between the plasma membrane and the vitelline membrane. So when, of course, the acrosome reaction takes place, there is a change in the electric potential of the oocyte membrane. And this one is what we call the first lock. This one is followed by the cortical granules, the cortical granules inside. They will discharge their content. And when they discharge their content, this is what we call the cortical reaction. And they are going to create an osmotic gradient that will draw the water between the plasma membrane and the vitelline membrane. The two membranes are lifted away and the vitelline membrane hardens, what we call now fertilization membrane. This one does so to block polyspermy. While the sperm tail is lost and disintegrates, the nucleus expands and is now known as pronucleus. So the other two membranes are going to be lifted away. And when those two membranes are lifted away, now the vitelline membrane is going to harden. When the vitelline membrane hardens, that is what we call fertilization membrane. And that fertilization membrane blocks polyspermy, not allowing more than one sperm nucleus entering into the oocyte. So as the sperm nucleus enters, the tail remains outside. It disintegrates. And when the nucleus now goes inside, it will expand. Now that nucleus is referred to as a pronucleus. Entry of the sperm stimulates the completion of the second meiotic division of the secondary oocyte, forming the second polar body, which disintegrates. And an egg, the haploid male and the female pronuclei fuse to form a diploid zygote, which divides immediately by mitosis to form two diploid cells. So what is going to happen is that when the sperm enters, it stimulates the secondary oocytes to form the secondary polar body. So it completes the second meiotic division when the sperm head enters. And now the haploid male nucleus and the haploid female nucleus, they will fuse together to form a diploid zygote. Now this diploid zygote will undergo what we call mitosis to form two diploid cells. Part B. Outline the extraembryonic membranes associated with the human fetus. The first membrane is the chorion. It completely surrounds the fetus and is the fetal contribution to the placenta. Then there is another membrane called the amnion. This forms a fluid-filled amniotic cavity that cushions the fetus from shock and mechanical damage. The third one is the yolk sac. This contains little or no yolk. It is the temporary site for red blood cell formation. Then there is the allantois. This one is derived from the embryonic hind gut, and it contributes the blood vessels that form the umbilical cord. (32) What are the main features of reproduction in birds? Part B. How are birds suited for reproduction on land? C. Compare the embryo development in birds and mammals. And D. State the form of parental care provided by the mammals. What are the main features of reproduction in birds? 1. Fertilization is internal, meaning it takes place internally. And mating is preceded by elaborate courtship display. The hard-shelled eggs, cladle, amniotic egg, are laid in an external environment. The eggs are incubated, usually by the mother, as the embryo develops. The newly hatched young ones, they are fed and cared for by the parents. Part B. How are birds suited for reproduction on land? 1 is the production of a hard-shelled egg for protection from mechanical damage. 2. Fertilization being internal. This avoids the drying up of the eggs and wastage of gametes. The newly hatched young ones, these ones are fed and cared for by the parents. For example, net building, brooding, and many others. The zygote develops within the amniote chlamydic egg, which provides the embryo with fluid-filled cavity in which it develops on land. Part C. Compare embryo development in birds and mammals. We can start with the similarities. In both, the embryo is surrounded by extraembryonic membranes, which develop from tissue outside the embryo. In both, embryo is cushioned in a fluid-filled amniotic cavity. Embryo development is preceded by internal fertilization in birth. Allantois is involved in gaseous exchange. There is protection from predators. Let's now look at the differences between the embryo development in birds and embryo development in mammals. Embryonic development in birds, the yolk sac is well developed and nourishes the fetus. Whereas in the mammals, the yolk sac poorly developed, since the fetus derives nourishment from the mother. In birds, allantois is a depository organ for nitrogenous wastes, like uric acid. In mammals, nitrogenous waste like urea diffuse into the maternal blood. In birds, the embryo is protected from damage by an outer shell. In mammal, the outer shell is lacking around the developing embryo. In birds, the yolk sac transfers digested food to the embryo. In mammal, digested food is transferred by the placenta. In birds, allantochorion is lacking. In mammal, there is a developed allantochorion. Part D. State the forms of parental care provided by mammals. There is feeding. They feed the young ones on milk from the mammary glands. They provide shelter to the young ones, and also they train the offsprings. State the physiological and morphological adaptations of flowers to pollination. Part B. Explain the mechanism which limit inbreeding and promote outcross in plants. State the physiological and morphological adaptation of flowers to pollination. Let us start with the physiological adaptation for wind pollination. The stamen and the carpels mature at the same time. Hermaphrodite flowers may not open the petals. Hermaphrodite flowers, they may remain underground. Pollen is released onto the stigma by matured anthers before the petals open. Or pollen is compatible with the tissues of the style, thus allowing their germination. Let's look at the morphological adaptations for wind pollination. Flowers are reduced and inconspicuous. The stamen are situated above the stigma, and the anthers is close to the stigma. The stigma often coiled to touch the ripe anthers. The style and the filaments coil on one another. The flower failing to open, for example, in Commelina. Let's now look at the physiological adaptation for insect pollination. The stamen ripens before the carpel, which we call protandry, like in Slavian dendinites and dandelions, or the carpels ripen area, which we call protagony. There is self-incompatibility due to chemicals. This prevents germinating of the pollen on the stigma of the same flower, which we call self-sterility. It is genetically controlled by self-incompatibility genes. For example, in pear pollen only becomes functional if the stigma surface on which it has a different genetic composition. Let's now look at the morphological adaptation for insect pollination. Dioecious flowers. They have either stamen or pistil. Stamen situated below the stigma. Hermaphrodite, those are bisexual flowers, contain both sex organs. Production of nectar. This attracts the insects. Hanging of the stamen or the whole flower downwards so that the falling pollen drops clear of the plant. Part B, explain the mechanism which limits inbreeding and promotes outcrossing in plants. Dioeciousness. All flowers on the plant being either male or female, like for example in purples. Monoecious. Having separate male and female flowers on the same hermaphrodite plant. Explain the mechanism which limits or promotes outcrossing in plants. Heterostyle. Different style length. Structure of the flower, for example, when a stigma is protected from coming in contact with its own pollen, or stigma being taller than the others. Dichogamy. The stamens ripen before the carpel, protandry, or the carpel ripening area, protogamy. Part B. Explain the mechanism which limit inbreeding and promote outcrossing in plants. Self-incompatibility due to chemical prevent germinating of the pollen on the stigma of the same flower. Thirty-four. Outline the differences between internal and external fertilization. Part B. Explain the evolutionary significance of internal fertilization. Part C. State the role of Sertoli cells in maintenance of sperm motility. Outline the differences between internal and external fertilization. Internal fertilization, there is production of large number of gametes, which is unnecessary. External fertilization, production of large number of gametes is very essential. In internal fertilization, offspring could be protected within the mother's body. In external fertilization, offspring are usually not well protected. In internal fertilization, offspring can be allowed ample time to develop and get born at a later stage of development. External fertilization, offspring are often born when too young and helpless. Part B. Explain the evolutionary significance of internal fertilization. Internal fertilization has led to the production of offspring at a later stage of development, capable of surviving and adhering to variable environmental changes. Two, internal fertilization has led to evolution of organisms that can reproduce in a variety of environments without necessarily returning to water. Internal fertilization has led to evolution of organisms that produce less gametes, since internal fertilization is associated with greater chances of reproductive success. Internal fertilization led to the evolution of reproductive strategy involving embryo being retained in the mother's body, where it is provided with appropriate conditions for growth. For example, terrestrial invertebrates like insects are involved in oviparity, laying eggs with little development, ovoviparity, laying well-developed eggs, and viviparity, extra-embryonic development. State the role of Sertoli cells in maintenance of the sperm motility. It is a site for further development of spermatids into well-developed sperms. It provides nourishment to the developing spermatids. It secretes a fluid that fills the lumen of the semiphyllous tubules that phagocytose foreign particles. It secretes inhibin that inhibits the secretion of follicle-stimulating hormone and luteinizing hormone to reduce the rate of spermatogenesis. It forms a barrier, prevents the entrance of harmful substances into the tubules that can interfere with the spermatogenesis. It prevents the sperm from passing out of the tubule into the blood, where they could stimulate an immune response. Tight junctions between Sertoli cells also form compartments to separate the sperm cells in various stages of development. 35. Describe the structure of the mammalian placenta. Part B. Explain the series of events that occur immediately after fertilization till the formation of the placenta. Describe the structure of the mammalian placenta. The placenta is a thick, large, long, discoid structure. It consists of cells derived from the fetus and the mother. The fetal part of the placenta consists of the chorion and finger-like projections known as the chorionic villi. So when you look here, we have the chorion plate, and then we have the amnion, and we have the umbilical cord. The maternal part of the placenta consists of the endometrium, maternal uterine tissue. The chorionic villi grow inside the endometrium. A space known as lacuna exists between the chorionic villi and the endometrium, which is supplied with network of arterial blood vessels of the mother. Arterial and venous blood vessels run between the fetus and the endometrium is a tough long structure called the umbilical cord. The umbilical cord is covered by cells derived from the amnion and the chorion. This is our umbilical cord. And we're saying the umbilical cord contains arterial and venous blood vessels that run between the fetus and the endometrium. Explain the series of events that occur immediately after fertilization till the formation of the placenta. The zygote now begins a series of cleavage divisions, which are rapid cell divisions without cell growth. As a result, each of the resulting cells, called the blastomeres, contains substantially less cytoplasm than the original zygote. Successive cleavage division result in a small ball of cells called a morula. As cell division continues, liquid-filled morula and pushes the cells out to form a circular cavity surrounded by a single layer of cells. This hollow sphere of cells is called a blastula, and the cavity is called the blastocoel. Formation of gastrula or gastrulation occur when a group of cells invaginate, that is, move inward into the blastula, forming a two-layered membrane, with the opening from the outside into the center cavity. A third cell layer forms between the outer and the inner layer of the invaginated membrane. These three cell layers of the gastrula, the ectoderm, mesoderm, and endoderm, outside, middle, and inside layer respectively, are the primary germ layers from which all subsequent tissues develop. Extraembryonic membrane development begins with the chorion, which is the outer membrane. Implants into the endometrium. Later, the chorion together with the maternal tissue form the placenta, which is a blend of maternal and embryonic tissue, across which gases, nutrients, and wastes are exchanged. Thank you very much for participating in reproduction in living organisms. Questions 31 to 35. Please do like, share, and subscribe.